Hello, everybody. These are the key words we're going to go over. Antibody, antigen, immunoagglutination, and fluorescent particles. Okay, so the first one, antibody. What is an antibody? I like to think of antibodies as the little defenders in our body. That blue, strong circle guy is an antibody. They are defending the people. And animals produce antibodies as well. That's why there's a pirate cat. So what do the antibodies defend against? Antigens. These are chemicals, bacteria, and viruses. These come from outside sources, and if they get into your body, that's when you need to produce the antibodies in order to protect yourself. Over there, Pirate Cat clearly has a little spiky red virus, but luckily some antibodies have gone and attacked it so that it won't get sick. So what happens when an antibody and an antigen clump together? Well, this is called immunoagglutination. This is a long, fancy word. Immuno means immune, like immune system, which is when your body defends against stuff. And agglutination is just a long way of saying clumps. So we already saw an example with a human and a cat, pirate cat. But what about foods, surfaces, and water? Well, all of these things can still have antigens on them. But we know that a human and the cat, so animals, they produce antibodies to defend. But how do we figure out if a food, surface, or water source has an antigen? This is the big question that people are trying to figure out, because if you can find where the antigen are, you can help prevent getting sick. So let's take a look at a water source. Here we've got 2 cups of water. The left one, clean, no antigen. The right one, it's got a red spiky guy, so it's got an antigen. How would we tell if which glass of water has the antigen in it? Well, what researchers like to do is they prepare 2 sources with the antibodies in them. And then you pour the water in, and the one with the virus, the antigen, that's the one that's going to have immunoagglutination. And then finally, the big problem is, how are you actually going to see if immunoagglutination is happening? Well, these things are really, really small. So for example, if we're looking at a strand of purple hair in the microscope, it's pretty small, but you can still see it in the microscope. But what happens if we want to take a look at an antigen and an antibody in a microscope? Well, actually, these are so small, you can't see them. So we need to get a little bit clever for a way to see them in a microscope. That's where fluorescent particles come in. I love fluorescent particles. These are so cool, and they really help us out a lot. So you can think of a fluorescent particle like a little star that glows. And then what scientists do is they attach the fluorescent particle to the antibody so that when you take a look at it in a microscope, look, there's a tiny little green dot. It's still not very big, but at least we can see it now because we can look for the glow from the fluorescent particle. Let's take a look at what would happen if we had some immunoagglutination with the fluorescent particles attached to the antibodies. Okay, so we have 2 little antibodies. They've both got their fluorescent particle attached, and then we're going to add our water source with the antigen. Here it comes. And look, in the microscope, it's a bigger, glowy clump. And that's how we find immunoagglutination. Woo!